EBSCOR, the experimental program to stimulate competitive research, is a National Science Foundation grant program. The goal of EBSCOR is to strengthen science and engineering research and education throughout the United States, and to enable states like Wyoming to be nationally competitive in these areas. EBSCOR aims to improve scientific infrastructure in Wyoming and build science, technology, engineering, and math research capacity across the state. One way Wyoming EBSCOR helps strengthen science research and education is through student programs such as the Summer Research Apprentice Program, also known as SRAP. SRAP is an intensive 6-week paid research experience for high school sophomores, juniors, and seniors aiming to attend college. It offers students a hands-on research opportunity in science labs across the University of Wyoming campus. Students work one-on-one with a faculty mentor throughout their time in the program. SRAP not only provides students with a paid internship and real research experience, but also helps prepare them for college life.
Miss Esmeralda Soto from Denver. She will be presenting her topic, Cathepsin K knockout attenuates aging-induced liver damage in mice.
Like Leo said, my name is Esmeralda Soto, and I did have Cathepsin K knockout can attenuate to age-induced liver damage in mice. So as an intro, we all know that the liver is a vital organ that can help control most chemical levels in the body and also takes away damage. And in aging is associated with damage in the liver, and it can help with apoptosis and fat in liver. So apoptosis is basically a programmed cell death that the cells will go through in order to help the surrounding cells to function better, and apoptosis contributes to age-induced organ malfunction. So this is the basic process that apoptosis goes through. So from the regular cell, it goes, the chromatin is this black thing that goes around the nucleus, so it's going to condense and it's going to get smaller and go to the edge of the nucleus, and then the membrane is going to start deforming, and then eventually it's going to cause the nucleus to collapse. So all the DNA and everything that's in the nucleus is going to be in the cell until eventually the membrane takes apart all those parts in the cell, and then with the lysosomes are going to start eating away the membrane and all the parts of the cell until the cell dies. So this is the basic pathway of apoptosis, and in this experiment we use both a pro and anti apoptotic proteins in our experiment. So Cathepsin K, it's a protease that is going to play it's right now it's used to play a major role in bone and bone resorption, and it also is involved in the pathogenesis of cardiovascular disease through activating apoptosis. So in this experiment, we hypothesized that by using Cathepsin K in Cathepsin K knockout, we can restore damage from the age-induced liver and inhibiting apoptosis. So we used two major types of mice. We used the wild-type mice, which are just regular mice, and then mice that were used with Cathepsin K knockout. And then within those two groups, we had old mice, which were 24 months, and young mice that were 6 months. And then we also did an H&E staining and oil red O staining for each, and then a Western blot analysis. So this is the basic procedure for the Western blot analysis. So using these gels, we would load the protein and then put it into a running buffer with electricity, and then the electricity would go down and it would separate the proteins depending on their weight. And then after, we would have this gel and we could use it to put it into sandwich-like things that were to transfer the gel proteins onto a membrane. and then that we can use to detect the bands of protein. So some of our results, we found that cathepsin K knockout attenuates aging-induced body weight and liver weight gain. So for the wild-type mice, we found there's a major difference in both the body weight and the liver weight compared to the cathepsin K knockout mice, which there wasn't much difference in both the body and liver weight. This is the results of the oil red staining. The oil red staining was used to find and indicate lipid accumulation. So as you can see, it's really hard to see, but there's red dots on this wild type old mice. So that means that there's a lot more lipid accumulation on this wild type mice compared to having barely any on the cathepsin K knockout. So we could assume that from this, that the cathepsin K knockout could help decline the amount of lipid accumulation. And our results for the H&E staining. The H&E staining would just stain the basic structures of the cells in the liver sample. So these white parts are basically vacuoles or empty stays and fat accumulation. So in the wild type old mice, there is a lot more empty space and cell death compared to the cathepsin K old mice. And then in the cathepsin K young mice, there is barely any, even though there wasn't any in the wild type also. So we could assume from this one too that the cathepsin K knockout can decline the age-induced morphological changes in the liver, and that's also good because less death in the cells means that the liver samples the liver can provide more abilities for a longer period of time. So this is our results for the Western blot, and we found that the cathepsin K knockout attenuates liver AKT and AMPK activity changes induced by the aging. So we can see that here these bands are a lot darker, so that means compared to the cathepsin K knockout, so that means that there's more apoptosis, which is bad for the liver. And then to get these graphs, we basically just found the average molecular weight for each of those and found the average to make the graphs. And similarly, we found that using these two, one is a pro-apoptotic and one is anti-apoptotic. So this one, having it be darker means that there is less apoptosis happening, more apoptosis happening because it is just wild type of cathepsin K. And here we have an error, which I don't know what happened, but and then for this one, it's the opposite. So if this being darker means that there is less apoptosis, so that's also good. So for summary, we indicate that liver accumulation and the morphological changes in aging were all corrected by the cathepsin K knockout, and that apoptosis was activated and aging possibly to suppress AMPK phosphorylation, which is also rescued by the cathepsin K knockout, and that AKT phosphorylation was elevated in the aged liver, and the AMPK phosphorylation was suppressed in aged liver, and that was also rescued by the cathepsin K knockout. So pharmacologically, inhibition of cathepsin K can help treat liver diseases in the elderly. So technically, it could, if we had the technology, could help liver diseases. But with how the technology is today, it's really hard to change the genetics of people. So right now, it's not possible to use this cathepsin K knockout to help restore damage in the liver. So in conclusion, we can see that with aging, it could activate AKT, which will go into inducing liver damage, but then it would inhibit AMPK, and that inhibits apoptosis, which is linked to liver damage. But then by intimidating both of these with the cathepsin K knockout is going to slow down apoptosis, which is going to help with the liver damage. So acknowledgements, I want to acknowledge Wyoming NSF's board for funding the program, my mentor and graduate student, Dr. Anon, for having patience with us. My professor, Dr. Sri Dan, for taking us in, even though he had a lot of stuff to do. My lab partner and roommate, Danielle, for putting up with me 24/7. And my fellow SRAPers and SRAP staff for making this an unforgettable experience, and Lisa for just coordinating everything. Question? Thank you.